So good morning, everyone. Welcome to this session of the Sci Cafe that is organized by the International Relations Office of the Hellenic Mediterranean University and the European University of Athena. It's a great pleasure to have with us Professor Guglielmo Lanzani. He's a professor in physics in the Department of Physics in Politecnico di Milano and also a coordinator of the Center for Nanoscience Technology of IIT in Milano. A few words about his career. His basketball career, he will tell us more. But regarding his professional career, in 1987, the year that the Greek national team won the Euro basket, he accredited the bachelor degree in physics. From 1989 to 90, he was a visiting scientist in Utah, US. And then in 1992, he acquired his PhD in chemical physics. From 1994 to 1999, he was an assistant professor at the University of Sassari. And in 1999, if the data are correct, he joined the Politecnico di Milano as an associate professor. His research interests include the nanostructure and molecular materials using the photophysics for applications in energy, biophotonics, neuroscience, and medicine. His work has received more than 17,000 citations, and his age index is 70. One of the main focus of his research is the light to enhance the light sensitivity in living cells, tissue, or organisms for photocontrolling their functions by developing new actuators. Today's talk is entitled Cell Opto-Stimulation by Intramembrane Molecular Switches. And as you know, our rector and the rest of the dear guests, they have mentioned, we are very honored to have you, Guglielmo, to this Sci Cafe session. And thank you very much for your kind acceptance. Thank you.
Thank you, Kostas, for the nice introduction. And thank you, everyone, for being here. You're very kind. It's my honor, actually, to be here. I was thinking that we should have a Mediterranean conference, maybe. There was, in the past, there was a common language for the Mediterranean countries, what's it's a curious thing. So we have a common past. Anyway, I'm going to talk about the research that is highly interdisciplinary and multidisciplinary.
I'm sorry, Guglielmo, you are sharing not the full screen, but the other screen.
Ah, sorry.
So if you switch, if you go to the display settings.
Yeah. Now I swap. No. Like this, you
yes,
see.
Yes. Thank you.
Ah, okay. Now I was saying that the research is highly interdisciplinary, so there is a lot of people involved with different competencies, expertise. And so here you see there are medical doctors, neuroscientists, biologists, chemists, a lot of people. And that's a challenge. I mean, additional to others when you move into this kind of research that should be considered. Now, introduction, the ultimate goal of what we do is to establish the symbiosis between artificial and living matter or device, so the abiotic coupling. Here you see a number of outcomes. It could be in regenerative medicine, in prosthetics. I use the laser pointer. For instance, this is the liquid retina project we are doing. It can be coupling to the brain. This thing is very nice. It's a soft robot which is powered by muscle cells, which are activated by optogenetics. I will show you that we do similar things, but without optogenetics. Now, in order to control with light the life and physiology, we need to introduce phototransducers. So these are functional interfaces that allow to establish the abiotic-biotic coupling. So typically what we do is we have this phototransducer, they absorb light and they transform it into bioelectricity, giving a signal. So all the cell that has this phototransducer can be switched on and off by light. So give us a handle to control. The target for our action is the membrane. So that to say, we don't go inside the cell because that's changed quite a lot the way things occur. So we target we aim at the membrane either say in contact or even now I will show you inside the membrane. Now, why the membrane? Well, the first experiment that we did many years ago was to grow on top of a photovoltaic polymer. This is a polythiophene, which I think everyone knows is a famous workhorse for photovoltaics. So we grew on top living cells. That could be HEK cell lines or neurons, whatever. We saw that they like to be there, so the polymer is not phototoxic. And then we discovered that shining light was absorbed by the polymer because the cell is transparent, and then it was somehow reacting on the cell and measuring the membrane potential. We could see a change. So more in detail, this is the experiment that we did, the first experiment. You see that we shine light, and the membrane potential, which is usually negative, and we leave the resting potential, was enhanced. So there was a depolarization, was less negative, and then it was decay, and there was an overshooting and coming back to equilibrium, switching off the light. So the blue, say the rectangular here, this is the light. So in this way, using the polymer as a transducer, we were able to introduce a effect of light, which in this case, we assign simply to a photothermal effect. So the light comes, it increasing the local temperature, and the local temperature is changing the capacitance of the cell. This is a measurement, so it's not say a model, it's something that has been measured. So the capacitance of the cell is changing. Now why? Well, the photothermal effect is mainly associated to a change in the geometry of the membrane. And actually, what you would say is, well, I mean, what one aspect is an expansion, but an expansion, like if the thickness is increased, as you know, in the plain capacitor model, it would be the other way around. Indeed, what happens is that when you shine, say, when you increase the temperature, there is a phase transition to something I discovered later that was very well known by the people working in the field. This is 1980, they knew. But of course, in 2015, I didn't know, so I thought it was a surprise that there is a phase transition and the membrane becomes thinner. Now, if you assume the elastic model mechanically, then it is thinner, but the area is larger. And so there are these two effects, both go to increase the capacitance. And then using molecular probes, I don't go into detail here, but there is a paper there. We also discover that the water goes into the membrane. That is an additional effect that is changing the electric function. Now, if you change the capacitance, you may expect, you understand, I will show you later even some simulation, but obviously if you change the capacitance and there is a potential difference across, there will be currents and there will be electrical effect following. Okay, now this was the inspiring initial work. Then I borrow an idea from nature. So essentially in nature, there are two main strategies to harvest energy from light. One is the photosynthetic strategy, which is a special pair of charge separation. The other one is photochromic. So in our retina, for instance, retinal undergoes isomerization 1,000 times per second or less. And these start all the cascading process that give us the photon detection signal that goes to the brain. So the way that nature interacts with light is with the photochromic molecule that is initially storing the energy of the light. So I took this idea and I asked my colleague, friend Chiara Bertarelli to synthesize for me a molecule. I asked her a molecule that photochromic with the largest possible steric hindrance and steric change. And that was amphiphilic. Why? The idea was to have something that it goes into the membrane. So it should be amphiphilic because if it is polar and apolar, it goes to the membrane and it stays there for a while. And the other idea was if it's changing shape and this is changing the volume, there will be an effect on the membrane. Indeed, say simulation done by Luca Maragliano, these are molecular dynamics. They show that this membrane, these molecules goes into the membrane. So the thing is working. Of course, I mean, it's not a huge surprise because I was copying the cell mask, which is a staining dye commercially available to see the cell membrane. So I was copying them. But anyway, it was working. So we are happy. The membrane, the molecule goes into the membrane. And you see here that this is a picture for neurons. You see that the molecule is decorating and it's going inside actually the membrane of the neuron, and it stayed there for 100 hours, say, which is, I mean, it's not forever, but it's long enough for in vitro measurement for sure. Now, next point, we did some photophysics, which is my original job. And so we studied the isomerization of the molecule, because when you change the chemical structure, you never know what is going on. And what we found out is that indeed the molecule behaves properly. So you see that when we shine light, the absorption of the trans isomer goes down, and the new peak, new absorption here and here small, it's appearing associated to the cis isomer. And here you see the dynamics in time, and you may notice that this is a second time scale. So what I show you here is the population process, collective population dynamics. So that means it's the number of molecule average that are in the trans, from trans go to cis state. Of course, it's a slow process because it comes from the equilibrium between isomerization and back transfer from the cis back to the trans. And the rate constant is simply the absorption. So it's the cross section. So it's a normalized cross section. So the faster you absorb light, the more molecule turn from one to the other isomer. You can do the same measurement in photoluminescence, which is important because in biology, say in living sample, it's difficult to measure absorption, but it's easy to measure photoluminescence. So we look at photoluminescence, it's the same thing. Because the trans isomer is emitting and the cis is not emitting, when there is isomerization, the luminescence goes down. So you can follow the same process by looking at luminescence. Now, what is the time for the single molecule? Well, obviously, a single molecule is turning in a much shorter time. And that's what we measured here by doing a pump probe experiment. Here there is a trick. You need to refresh your sample. So you need a new molecule for each pump pulse coming for the measurement. Which means that we have to circulate the sample. Okay, once you do this, that's okay, that's enough. So you circulate, and then you are not accumulating the cis molecule, so you can follow the process of trans to cis isomerization. And what you see here is that it takes few picoseconds, actually time constant is 6 picoseconds, to turn a trans conformation into a cis one. And then the cis leaves for nanoseconds. Okay, then this was measured in DMSO, which is the best solvent of the molecule. And then we say, let's try in water, because water is what we could use for biological application. And here there was a bad surprise. In water, nothing like that is occurring. You see that you do not form the absorption of the cis isomer, which is the red component here. This is the global fitting of the overall pump probe measurement. But you see in water, there is just some relaxation, and you just have something that is decaying and red shifting, but there's no isomerization. The interpretation is like this. So in the DMSO, in the good solvent, we get the conical intersection to go to the cis isomer, the usual behavior. But when you go to water, there is a aggregation. You form dimers or even larger aggregates, and they form hexamers. So the wave packet reached the conical intersection, but there's no more chance to go to the cis isomer. What happened is that it's locked in the hexamer. The luminescence is stronger, but there is just decay back to the ground state. So that apparently was bad news. But then studying the molecule in different media, we observed that you see the photoluminescence spectrum is when you go from water, which is the red one, to the surfactants or even the cell membrane, it's going back toward the good solvent spectrum. So you see that the green spectra, which come from the surfactant liposomes or the cell membrane, is something in between water and a good solvent. So this suggests that when the molecule is in contact or can go inside the membrane, the cell membrane, it is recovering the isolated molecule behavior. And indeed, when we did the measurement, we saw that you see, looking at photoluminescence, there is again the isomerization. Since the decay of the photoluminescence will be formed cis from trans, sorry. And you see DMSO is a larger signal, but in SDS, which is this mimicking medium of the membrane, there is isomerization. Now the signal is small because not all the molecules are inside the liposome made by SDS, so many are in water, so they do not turn, but the process is back. And indeed, when we did PAM probe, that was very, very clear. You see that we rescue the behavior, the good behavior. So when we measure the molecule in the membrane mimicking medium, the trans decay and the cis form. And you see here appearing the absorption of the cis isomer. So it's behaving like the good solvent. So that's good news. And I can show you this, that's very instructive, that in a cell, when you shine light, you see that there is this dark line here, right? So only the molecule in the membrane can isomerize, and so you can switch off photoluminescence. So that's a clear indication that the membrane is protecting the molecule, and it is allowing isomerization, which is otherwise lost when the molecule is in water. That means when the molecule is outside the cell or totally inside the cell, it's not working. But when it is in the membrane, it is still isomerizing. So that's a good thing, because it suggests we can have an effect. And indeed, we did the following experiment. You see here a cartoon. So the molecule is in the membrane, and we shine light, and we measure with patch clamp technique, the electrophysiology, the change in the membrane potential. And what we find out is that indeed the membrane potential is changing. You see that it becomes more negative. So there is hyperpolarization of the membrane potential following light. When we switch off light, the membrane potential, the rest goes back to the resting value. And there is a small overshooting, a little bit of rebound on the other side here. The same thing is the light lasts longer, say 200 milliseconds, but there is depolarization. And then the resting potential go back, and there is some noise here towards depolarization. You see that the effect is following. There are a few points here. These are taken from electrophysiology, changing the LED source. But anyway, you see clearly that there is a peak, which is more or less where the molecule is absorbing. So actually, we are seeing something that is associated to the molecule. So the membrane, say the molecules stay in the membrane, and we shine light, and we can change the potential. So that's interesting for application. And what we saw mainly was hyperpolarization. Then we went to neuron. Now, neurons are much more interesting, and they are much more complicated. So they have more ionic channels and functions. And indeed, there is something that is very strange and unexpected. You see here is the same thing. You see that there is initially the same behavior. So we shine light, and the neurons, where our molecule is inside the membrane, hyperpolarize. So they become more negative. That means we are switching off the neuron. It's not firing action potentials. But then it goes back, and it goes all the way on the other side. But now the rebound, the signal here the overshooting is much larger. Is almost as large as the initial hyperpolarization. So there is a very large depolarization. Now this phenomenon has something to do with the biology of the physiology of the cell, and probably is due to the anode braking mechanism. This is a strange mechanism that is occurring in biology. That means when you shine light and you reduce the hyperpolarize the cell, then for a strange opposite thinking, say, it is easier to depolarize. Because when all the channels are closed, then the threshold for reopening is lower. That's the anode braking mechanism. It's one of these mystery of biology that is difficult to understand. It's difficult to catch, but anyway, to capture the meaning. But that's what happened. Now, what happened in neurons then when we look at the behavior in a network, and that's the experiment that I show you. Is here you see we shine light. There is hyperpolarization. There is depolarization. And now, most of the time, before I was showing you a few that we were not firing, most of the time there is firing because depolarization is large enough. So the trace below, the red trace here, show you that with light, we can start action potentials because they are associated to depolarization. Of course, if you do the same thing, just adding the MSO, which we are still using, so what we inject is a small fraction of the MSO in water, and this is added to the culturing medium, and then we wait a few minutes, and then the molecule go into the membrane. Now, the DMSO, of course, has no effect, so the control is flat, but now we have a tool in order to photo-stimulate neurons. So that can be very interesting for many applications regarding disorder of the central nervous system. Of course, in principle, can be also useful for the retina, even though the short time, permanence time is obviously not a good news. Now, we show as a proof of concept that you can also measure something in vivo. So this is done in the brain of a mice in which we introduce our molecule, and then we come with a microelectrode and a fiber optic. And the optical fiber is used for stimulating molecules. The molecule absorbs the light, generate bioelectricity, give the signal, and then the multi-electrodes are recording the local field potential. So it's like the electrocardiogram, it's not patch clamp. But you see here clearly that when there is the molecule, there is a signal. So even in vivo, in principle, our molecule is working. Now, what about other applications? Well, for instance, we are interested in bacteria for many reasons. A bacteria is a small machine that you may think to use as a component in a device to develop living technology. So that's an interesting thing. They are used as electrodes for electron transfer, and we want to develop some photonic application. Now, the first thing we notice is that our molecule is also sitting in the membrane of the bacteria, and you see here that the zeta potential, the Z potential of the bacteria is reducing, which what we assign to the interaction of our molecule with the electrical charge of the membrane. So that tells us that indeed our molecule is sitting in the bacterial membrane. And now what is interesting is that even in bacteria, you see if we measure the dynamics, the photoluminescence, we have a sign, this lowering here in the photoluminescence sign of isomerization. And this is an optical measurement of action potential, say of membrane depolarization, and you cannot do electrophysiology in the standard way of bacteria, but you can put voltage sensitive dyes. And these voltage sensitive dyes show that there is an effect on the membrane potential when we shine light. So in principle, we can also change the potential. We can depolarize the bacteria cell. Then we found a phenomenon that, I mean, I don't know what to do with it, but it's there. I mean, so I tell you. It's, so we were looking at the minimum inhibitory concentration. That means in the dark, if we add our molecule, is the gray, you see that we kill the bacteria. So the molecule is an antibacterial. So it is killed. So the bacteria population does not proliferate, does not grow if we add our molecule in the dark. Okay, now if we shine light, this antibiotic effect is much reduced. For instance, this concentration, 2.5 millimeter, that's it's a number, the concentration to start. You see that the population is not affected. Then only when the concentration is pretty high, it is killing. But the difference is always very clear. So when we shine light, the antibiotic effect is much smaller. And again, the phenomenon depends on the, I mean, it's clearly has the wavelength dependence that we expect from the molecule. What to do with this, I don't know, because I mean, it would be, maybe the other way around could be interesting, but to stop an antibiotic is not so clear what it's good for. But anyway, another thing we did is to study muscle cell, cardiomyocyte, or skeletal muscle cell. Because I don't like to kill animals for this, we say we are working on human-induced pluripotent stem cell. Now, don't ask me how they do this because I don't know. It's a magic thing. They take cell from the skin of a patient, for instance, a human. They reprogram back the cell to the pluripotent state, and then they differentiate the cell into a cardiomyocyte. That's, I don't know, it's amazing that genetic engineering or whatever it is, they say it's biotechnology. Then once you get the pluripotent, you can have different things, and so you can get cardiomyocyte. And those cells can be used for cell replacement, for drug screening, and to study disease, because something that's very interesting is that if you take the cell from a guy that is sick, that has some disease, you find the same disease in your cell here. So you can study, like if there is LA or there is other effect of the muscle cell, some problem there in the muscle cell, you find the same problem here because the genetic, of course, code is in the cell. So now we get these cells or we buy cell lines for skeletal cells and we grow them on oriented pattern. Now, if you grow muscle cell in an oriented pattern, at some point they combine together and they make large cells that become fibers, and they become the structure, the microstructure of the muscle. So they start to move macroscopically. First of all, in the single cell human-induced pluripotent stem cell sample, I show you here that when we add our photochromic molecule, we can induce action potential. And we can increase the contraction rate. These are moving cells, right? So they contract naturally, spontaneously. When we shine light, we increase the frequency of the contraction. So that means we have a control over the mechanical contraction of the cell. And this can be used to build a cyborg. And that's what Vito did, collaborating with the group in Harvard by Kit Parker, because we were not able to make the whole process. Now, you see here a comparison between two PDMS film. On one, there is no light stimulation. On the second one, we did light stimulation, and you see it is moving. So we can build soft robots, and we can obtain macroscopic movement driven by muscle cell. And well, this is a measurement of the calcium propagation wave, which is associated to the cell contraction. And you see, well, you don't see here, but I tell you that the propagation is the same if there is our molecule or not. So that means we are not affecting too much the fundamental, say, the physiology of the cell. Now you may ask me, what for? I mean, nice, but why should I have this thing moving? Well, this is an example of the same group in Harvard. That was something done in 2012. Here they are using electrical stimulation using electrodes. So it's just a proof of concept, because it's not very useful to have a big electrode applying at the solution. But you see what they did. They did a medusoid that can swim, and it is a cyborg. It is powered by muscle cell. I find this really amazing. It's similar to the fish I showed you at the beginning. And something else those people are doing is to use optogenetics. But optogenetics is interesting, but it has a number of limitations. So we like to do the same thing with our molecule with a non-genetic approach. All right, now very last, I want to discuss a bit the mechanism. So what we measure is a changing capacitance as well. So that's why I was showing you the changing capacitance at the beginning. Now you see, if you add the molecule, the capacitance in the dark with our molecule is increased. And when we shine light, it is reduced back to, say, towards the original value. So we modulate the capacitance. Here you see in time that we shine light, the capacitance is reduced, and then it goes back to the original value in the dark, in presence of our molecule. So there are two questions here to be answered. Why in the dark our molecule is changing capacitance, and why when we shine light, this change is reduced? And what we found out is that the thickness of the membrane is reduced when we add our molecule. And you see very clearly here, using AFM on an artificial lip membrane, that when you add, when the MSO is not doing anything, but when you add our molecule, you see the dark region are smaller, say lower levels, so they are thinner membrane spots. And it's very clear that this is happening. These are two kinds of membrane models, and you see both that there are a lot of dark spots which are associated to the thinning of the membrane. So there is a geometrical effect occurring. And now neutron scattering is giving the same outcome. And now it looks like the computer is stuck. I hope it will be back soon. Here it is. Say neutron scattering, I don't know this technique. My postdoc, Giuseppe Paternò, now is an associate professor, did this experiment. But neutron scattering is also showing that the membrane geometry is changing, and they suggest that the membrane become more disordered. It is, say then, the distance between the phospholipid is larger, and that means that the thickness is lower. So that goes in the right direction to be an increase in capacitance. And then we did a chemical approach. So we study a family of molecules which are similar but not the same. They are all good to stay in the membrane. So all these molecules are amphiphilic. They all have a azobenzene core. They all go into the membrane. But you see the effect on the potential membrane, the membrane, say the electrical potential, the voltage in the membrane is only good for these two family of molecules. Apparently, those molecules here are not active. That's quite surprising. I don't know why here there is a delay. Okay. Now, what is the mechanism which we think is going on? Well, the first thing I asked the guy that is doing simulation was to make a calculation of the thickness of the membrane in trans and cis isomer. And I was expecting the opposite behavior, that when it goes to cis, it is more steric interest, is say, enlarging the membrane. That's not what happened. So now, desperate because there was no effect, I asked him to study more than one molecule. So he did 2 molecules. And then he saw that they tend to dimerize. So if you put 2 molecules, there is a minimum of energy when they couple together, they dimerize like this. And so this means that the thickness is reduced. When you go to the cis isomer, the dimer is distorted and destroyed, and the thickness of the membrane goes back. So that supports an interpretation that is that there is a sort of molecular machine. The 2 photochromic molecules act to reduce in the dark the thickness, and this phenomenon is killed when we shine light. So geometrically, we can explain the phenomenon. Now, usually people at the point ask me, what are the evidence that there is this dimerization? That's theory. And I should say, I don't have many. The most obvious experiment, the concentration dependence, is not working. So we can change a bit the concentration and see the phenomenon, and nothing happened. And then at some point, we don't see anything anymore. So we are not able to have a clear behavior, a clear threshold for the thing. Now, the spectroscopy would suggest that if the molecule dimerized like this, there should be an excitonic coupling that make it an H aggregate. And the H aggregate should be associated to a blue shift of the absorption and red shift of the emission. And now, if you want to believe, if you want to see this, there is this effect. So the solid line is in the membrane, artificial membrane model, and the dashed line is in the solution. And you see that perhaps there is this effect. I mean, usually both say spectra should shift in the same direction. So it's a suggestion. Another thing that was done was to study artificial membranes. And when we studied these artificial membranes, they were able to introduce the membrane on one side and the other. And you see, if you put the membrane only on one side, the change in capacitance is much smaller. When you put it on 2 sides, it's much larger. So here you see the setup. They have an artificial membrane, and they add the molecule and they measure electrically with the electron in the 2 sides in the compartment. They see the change. I I mean, I don't know, this suggests indeed that if you come to both sides, there is a larger effect. I'm not sure it's really what we, I mean, is the real evidence. Last, I show you in a simulation using the equivalent circuit for the cell membrane, simplified. So here, this is reproduced ionic, the pump, and this is the conductance, and this is the capacitor that reproduces the membrane. So it's a standard equivalent circuit. And in the black trace is the change in capacitance that has been introduced in the model, and the red one is the change in the membrane potential calculated. So you see that in this circuit here, the membrane potential is indeed hyperpolarizing and then depolarizing. So it is reproducing very nicely what we observe in the HK cell. Of course, there is not the large overshoot that we see in neuron, but that's because in neuron is due to some other phenomenon which is not associated to the simple membrane dynamics that we study here. All right, so there are a lot of funding agency that should be acknowledged, but that's usually boring and you don't care. And so I say thank you very much for your passion to be here listening to me. Thank you very much.
Thank you, Guglielmo, for this very nice and inspiring talk. Let's give the floor now to the audience. I have some questions from curiosity point of view, but let's give the floor to the audience. If there are any questions from the audience, please, now is the time to direct them to Professor Lanzani. You can raise your hand, or you can switch on your microphone and direct the question to Guglielmo. Any question? So,
I have a question, actually.
Yes, Masoud, yes, please.
Thank you, Guillermo. It was very nice talk, and it was very inspiring and exciting research that you are doing. I have some question about the light that you are using. First of all, which kind of light you are using? Is it a CW light? Is it a laser light? Or is it a normal light? Or is it pulse?
It can be, sometimes it's a laser diode in the blue 470, 480 nanometer. Sometimes it's just an LED. It is, however, always pulse. You see that we have this duration, which is a few tenths of millisecond or hundreds of milliseconds.
Yeah, and for the measurement of the picosecond changes that you had in this range, what are you using to measure this time, small times?
Okay, that was the standard femtosecond pump probe setup. The pump there, for convenience, is 400 nanometers because they simple, the usual, easy to get, and the probe is white light continuum, so it's spanning the visible spectrum. So it's a standard pump probe experiment. So the resolution actually is a few hundred femtoseconds, which is even too much for what we observe.
Yeah.
The only trick is the circulation of the sample. So there is a peristaltic pump which is circulating the solution. Otherwise, you have a build-up at negative time, and you cannot measure the dynamics of the molecule.
Yes. And the wavelengths of the light, how sensitive is to the wavelengths, these experiments?
Well, it's better to be at close to the peak of the absorption of the molecule, which is in the blue region. So, but you saw in a few examples that we were tuning the wavelength, and it's following the absorption of the molecule. So it is still occurring even when you go to the threshold. But of course, I mean, it's proportional to the absorption of the molecule.
Okay, so you first measure this, of course, and then based on that, you choose your wavelengths.
Right. Yeah. Exactly. Yes.
Exactly. Okay, thank you very much.
Oh, my pleasure.
Thank you, Masoud, for the question, and Professor Lanzani for the answers. Any other question from the audience? There is a question from Dr. Kakavelakis from EPFL. The floor is yours, George.
Hello, thank you very much for the very nice presentation, Professor Lanzani. So I have a question regarding one experiment that I saw you used dimethyl sulfoxide DMSO in one of your experiment exposing DMSO with your cells. So my question is, I mean, isn't it this solvent, I mean, toxic solvents for your cells? And do you have any other, I mean, option to replace like DMSO with a more eco-friendly solvent? Thank you very much.
Yeah, right. Now, good point. Indeed, the first experiments we did, we were using DMSO in a small quantity. So it was a small part of DMSO in water, and then we were adding this suspension to the sample. And then DMSO, say probably the quantity was small enough not to be toxic, at least during the experiment. Then when I show you the family of molecules, because we were looking for something that can be used just in water, and indeed, we found out that you can just use water. So right now, we just use water. I mean, the molecule is not fully dissolved, so it's not the best solvent, but if you stay a low concentration, you can still manage with some steering, the usual trick. You can measure to have enough, as you say, dissolved molecules just in water, just to avoid DMSO, as you say. Anyway, I should say, just to be complete, that all the experiment has always been done using also the control test just with DMSO to be sure that it was not killing the cell, it was not toxic, it was not giving some effect on its own. So this was tested in the published paper. But that's a good point. I mean, I agree with you, we should get rid of DMSO.
Okay, thank you very much.
You're welcome.
Thank you very much, George, for the question and the answers from Professor Lanzani. Any other question from the audience? So let me ask you some naive questions now, Guglielmo. First of all, I would like to ask you, do you observe any differences regarding the electrical signal if you are going to use cancerous cells? And the other question that I have is like regarding the mechanical motion that you demonstrated. What are the figures of merit that control the amplitude of these motions using different laser parameters? For example, if you change the intensity or if you change the power. These are the questions that I have. Okay,
the first one, what you see, the cardio, you mean the comparison with the cardiomyocyte?
Yes.
Okay, but it looks like besides, let alone that the cells are very different, it looks like the effect of the molecule is the same. So actually in any kind of cell, because we have HEK, we have neurons, we have this C2, C12 also, is a model cell line for skeletal muscle cell, and cardiomyocyte, it's always behaving the same way. I mean, so we see the same thing. So it looks like really is this effect of the membranes. It makes sense because if it is the membrane, then it's something that is everywhere. Even the bacteria, which is surprising, behave the same way. Now, about the parameter, I think there is a lot to be studied. For sure, the amplitude of the response depends on the time constant of the cell, and the time constant depends on the conductance and, as you know, the capacitance. No? So the RC essentially is the response time of the cell. Now, first of all, there is a distribution of times. So cell to cell are different. You have to do statistics. That's biology. And the other thing is clearly that you can play around with the simulation and you see that, of course, if the pulse is too short or is too long with respect to this time constant, you get much larger or smaller effect. So that's something you can play around. We never did too much study on other parameters. So it would be interesting to see, for instance, with muscle cell, to see if we repeat a different duty cycle, what happened, because in principle, you can lock the contraction frequency with your stimulation. So far, we are not able, even though we send a train of pulses, the cell start to contract, but they increase the contraction, but it's not at the frequency that we are using for stimulation. I don't know why, but we are not able to lock the system. And so anyway, these are, I would say, next point to be explored. But that's a good suggestion, maybe. And
thank you, William. And something to make clear, because I did not get it, regarding the change of the capacity when you send, the light. Do you change the geometrical parameters of the capacitor, or you are changing the electric constant? Mm-hmm. I suspect that the geometrical capacity, you are changing the geometrical characteristics. Am I right?
True. Yeah, that's what I think. Yes, that's what I think. Now you may ask, what about the electric function?
Yeah.
That's a good point. I mean,
Because you are changing, you are creating something like you mentioned polymerization or deep. So you are changing something, no?
It's true. Yeah, no, you're right. And I mean, I still think the electric function is something, is a macroscopic property. It's not a single molecule property. So if the dimers are isolated, I don't think you can talk in terms of the electric function. Of course, if you have many molecules, in principle, for instance, in the dark, I cannot exclude that there is also a change in the electric function, because you get many molecules there and the polarity of this molecule is different. So I guess that there is a change in the electric function as well. With light, you're going to change a few spots. So not sure. There could be some effect also on the electric function. It's more difficult to be evaluated. I think the geometry is the larger part. Anyway.
Thank you for the question. So are there any questions? If not, cannot see any questions, let's close, the session. I don't know if my rector would like to say something at the end.
I would like to thank once more Professor Lanzani for this wonderful talk. I would like very much to see cooperation between the group of Professor Lanzani and our researchers in HMU. And I'm looking forward to meeting you from close, to see you also from close here in Crete. Thank you very much for this talk.
Thank you very much, Rector. Thank you very much, everyone, for being here. Really a pleasure. Thanks.
Thank you very much, Guglielmo. And you have an open invitation to come to Crete whenever you want. And
Sure.
Thank you. Thank you so much. Have a nice day, all of you, and see you next week.
Bye-bye.
Bye-bye. Thank you very much.
Bye. Kostas, can I have a short talk with you?
Just a moment, I will close the session and I will send you another Zoom link.
Okay.